Mineral Nutrition Part 1. In all living organisms, the basic needs are essentially the same. For their growth and development, they require macromolecules such as proteins, carbohydrates, and fats, water, and minerals. Methods to study the mineral requirements of plants. A prominent German botanist, Julius von Sachs, in 1860 for the first time demonstrated that plants could be grown to maturity in complete absence of soil in a defined nutrient solution. In a nutrient solution, this technique of growing plants is known as hydroponics. A number of improvised methods have been employed since then to try and determine the essential mineral nutrients for plants. The culture of plants in a soil-free defined mineral solution is the essence of all these methods. Purified water and mineral nutrient salts are required for these methods. Can you explain why this is so essential? A mineral solution suitable for the growth of the plant was obtained after a series of experiments in which the roots of the plants were immersed in nutrient solutions and wherein an element was added or removed or given the way in varied concentration. Essential elements were identified by this method and their deficiency symptoms were discovered. For the commercial production of vegetables such as tomatoes, seedless cucumber and lettuce, hydroponics has been successfully employed as a technique. To obtain the optimum growth, it must be emphasized that the nutrient solution must be adequately aerated. What would happen if solutions were poorly aerated? Essential mineral elements. In the soil, most of the minerals present can enter plants through roots. In fact, in different plants, more than 60 elements of the 105 discovered so far are found. Some species of plants accumulate selenium and some others gold. Some of the plants which grow near nuclear test sites take up radioactive strontium. Even at a very low concentration, there are techniques that are able to detect the minerals. The question is whether all the diverse elements of the mineral in a plant, for example gold and selenium, are really necessary for plants. How can we decide what is essential for plants and what is not essential? Criteria for essentiality. The criteria for essentiality of an element are given below. A. For supporting normal growth and reproduction, the element must be absolutely necessary. The plants do not complete their life cycle or set the seeds in the absence of the element. B. The element's requirements must be specific and by another element it should not be replaced. In other words, by supplying some other element, deficiency of any one element cannot be met. C. In the metabolism of the plant, the element must be directly involved. For plant growth and metabolism, only a few elements based upon the above criteria have been found to be absolutely essential. Based on their quantitative requirements, these elements are further divided into two broad categories: 1. Macronutrients and 2. Micronutrients. Generally, in plant tissues, macronutrients are present in large amounts. Carbon, hydrogen, oxygen, calcium, phosphorus, sulfur, potassium, nitrogen, and magnesium are macronutrients. Among them, carbon, hydrogen, and oxygen are obtained mainly from carbon dioxide and water, while the others are mineral nutrition are absorbed from the soil. In very small amounts, micronutrients or trace elements are needed. Micronutrients or trace elements include iron, copper, manganese, zinc, boron, molybdenum, nickel, and chlorine. There are some beneficial elements in addition to the 17 essential elements named above, such as cobalt, silicon, sodium, and selenium. The higher plants require them. On the basis of their diverse functions, essential elements can also be grouped into four broad categories. These categories are 1. Essential elements as components of biomolecules and hence structural elements of cells. 2. In plants, essential elements that are components of energy-related chemical compounds. 3. Essential elements that inhibit or activate enzymes, for example, for both ribulose bisphosphate carboxylase oxygenase and phosphoenol pyruvate carboxylase. Magnesium ion is an activator, both of which in photosynthetic carbon fixation are critical enzymes. During nitrogen metabolism, zinc ion is an activator of alcohol dehydrogenase and molybdenum of nitrogenase. 4. The osmotic potential of a cell can be altered by some essential elements. In the opening and closing of stomata, potassium plays an important role. In determining the water potential of a cell, you may recall the role of minerals as solutes. Role of macro and micronutrients. Several functions are performed by essential elements. In the plant cell, they participate in various metabolic processes such as permeability of cell membrane, electron transport system, maintenance of osmotic concentration of cell sap, enzymatic activity, buffering action, and also act as major constituents of coenzymes and macromolecules. Various forms and functions of essential nutrient element are given below. Nitrogen. Plants require this essential nutrient element in the greatest amount. It is absorbed mainly as nitrates by plants, but some are also taken up as nitrite ion or ammonium ion. All parts of the plant require nitrogen, particularly the meristematic tissues and the metabolically active cells. The major constituents of proteins, nucleic acids, vitamins, and hormones is nitrogen. Phosphorus. From the soil, phosphorus is absorbed by the plants in the form of phosphate ions. The cell membranes, certain proteins, all nucleic acids and nucleotides contain phosphorus, and it is required for all phosphorylation reactions. Potassium. Potassium is absorbed as potassium ion. The meristematic tissues, buds, leaves, and root tips in plants require potassium in more abundant quantities. An anion-cation balance in cells is maintained by potassium, and it is also involved in opening and closing of stomata, protein synthesis, activation of enzymes, and in the maintenance of the turgidity of cells. Calcium. From the soil, plants absorb calcium in the form of calcium ions. The meristematic and differentiating tissues require calcium. It is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella during cell division. During the formation of mitotic spindle, it is required. In older leaves, it accumulates, and also in the normal functioning of the cell membranes, it is involved. In regulating metabolic activities, it plays an important role and it activates certain enzymes. Magnesium. The plants absorb magnesium in the form of divalent magnesium ion. It is involved in the synthesis of DNA and RNA, and it also activates the enzymes of respiration and photosynthesis. Magnesium helps to maintain the ribosome structure, and it is present in the ring structure of chlorophyll. Sulfur. Sulfur is obtained by plants in the form of sulfate ions. In two amino acids, cysteine and methionine, sulfur is present and it is the main constituent of several coenzymes, ferredoxin and vitamins. Iron. Iron is obtained by plants in the form of ferric ions. In comparison to other micronutrients, it is required in larger amounts. In proteins, it is an important constituent involved in the transfer of electrons like ferredoxin and cytochromes. During electron transfer, it is reversibly oxidized from ferrous ion to ferric ion. It is essential for the formation of chlorophyll and it also activates catalase enzyme. Copper. Copper is absorbed as cupric ions. In plants, it is essential for the overall metabolism. Like iron, copper with certain enzymes is involved in redox reactions and reversibly it is oxidized from copper ions to cupric ions. Manganese. Manganese is absorbed in the form of manganese ions. In photosynthesis, respiration and nitrogen metabolism, it activates many enzymes. Splitting of water to liberate oxygen during photosynthesis is the best defined function of manganese. Zinc. Zinc is obtained by plants as zinc ions. Various enzymes are activated by zinc, especially carboxylases. In the synthesis of oxygen, it is also needed. Boron. Boron is absorbed as borate ion or tetraborate ion. For the uptake and utilization of calcium ion, pollen germination, membrane functioning, cell differentiation, cell elongation and carbohydrate translocation, boron is required. Molybdenum. Molybdenum is obtained by plants in the form of molybdate ions. It is present in several enzymes including nitrate reductase and nitrogenase where both of them participate in nitrogen metabolism. Chlorine. Chlorine is absorbed in the form of chloride anion. Along with sodium and potassium ions in cell, it helps in determining the solute concentration and the anion-cation balance. In photosynthesis, it is essential for the water splitting reaction, a reaction that leads to oxygen evolution. Deficiency symptoms of essential elements. Plant growth is retarded when the supply of an essential element becomes limited. The concentration of the essential element is termed as critical concentration, below which plant growth is retarded. When it is present below the critical concentration, the element is said to be deficient. In plants, since each element has one or more specific structural or functional role, and when any particular element is absent, plants show certain morphological changes. These element deficiencies are indicated by certain morphological changes and are called deficiency symptoms. From element to element, the deficiency symptoms vary, and when the deficient mineral nutrient is provided to the plant, they disappear. However, eventually it may lead to the death of the plant if deprivation continues. The deficiency symptoms shown by the plant parts also depend on the mobility of the element in the plant. Within the plants, for elements that are actively mobilized and exported to young developing tissues, the deficiency symptoms in the older tissues tend to appear first. For example, in the senescent leaves, the symptoms of deficiency of nitrogen, potassium, and magnesium are visible first. Biomolecules containing these elements in the older leaves are broken down and they make these elements available for mobilizing to younger leaves. In the young tissues, the deficiency symptoms tend to appear first whenever the elements are not transported out of the mature organs and are relatively not mobile. For example, the element like calcium is not easily released as it is a part of the structural component of the cell. To agriculture and horticulture, this aspect of mineral nutrition of plants is of a great significance and importance. In plants, the kind of deficiency symptoms shown include stunted plant growth, premature fall of leaves and buds, chlorosis, necrosis, and inhibition of cell division. The loss of chlorophyll leading to yellowing in leaves is called chlorosis. By the deficiency of elements nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum, this symptom is caused. Similarly, death of tissue or necrosis, particularly leaf tissue, is caused due to the deficiency of calcium, magnesium, copper, and potassium. An inhibition of cell division is caused due to the lack or low level of nitrogen, potassium, sulfur, molybdenum. In plants, if the concentration of some elements like nitrogen, sulfur, molybdenum is low, then there will be delay in flowering. You can see from the above that multiple symptoms can be caused by the deficiency of any element, and by the deficiency of one of several different elements the same symptoms may be caused. Hence, one has to study all the symptoms developed in all the various parts of the plant to identify the deficient element and compare them with the available standard tables. We must also be aware that to the deficiency of the same element, different plants respond differently. Toxicity of micronutrients. The micronutrients are required always in low amounts, and the deficiency symptoms are caused by their moderate decrease, and toxicity is caused by their moderate increase. In other words, the elements are optimum as there is a narrow range of concentration. In tissues, it is considered toxic if any mineral ion concentration reduces the dry weight of tissues by about 10%. Such critical concentrations among different micronutrients vary widely. To identify the toxicity symptoms are difficult. For different plants, toxicity levels for any element also vary. Excess of an element many a times may inhibit the uptake of another element. For example, the appearance of brown spots surrounded by chlorotic veins is the prominent symptom of manganese toxicity. It is important to know that with iron and magnesium, manganese competes for uptake and for binding with enzymes with magnesium. In shoot apex, manganese also inhibit calcium translocation. Therefore, deficiencies of iron, magnesium, and calcium may be induced by excess of manganese. Thus, symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Please share this video with your friends. Leave your comments below. Please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.